Today we're going to learn about DNA replication, but before we do so, we have to talk about the components of a nucleotide. A nucleotide consists of a 5-carbon sugar, deoxyribose in DNA and ribose in RNA, a phosphate group, and a nitrogenous base. There are 5 different nitrogenous bases: adenine, guanine, cytosine, thymine in DNA, and uracil in RNA. There are 2 strands in DNA. One strand runs from the 5' to 3' direction. The anti-parallel strand runs from the 3' to 5' direction. Basically, the subunits run in opposite directions, giving directionality to each strand. At the 5' end is where the phosphate group will attach, and the 3' end is where the hydroxyl group will attach. Helicase is the first enzyme in DNA replication. It comes through and it splits the 2 parental strands, breaking the hydrogen bonds between the nitrogenous bases. Then, after helicase does its job, another enzyme, single-stranded binding proteins, come and bind to each of the parental strands, keeping the strands from coming back together. This region right here is called the replication fork. It is simply a Y-shaped region where the parental strands of DNA are being unwound. Whenever the strands unwind, there is a leading strand and there is a lagging strand. First, we're going to talk about the leading strand, which is synthesized continuously in a 5' to 3' direction. The unwound DNA strands serve as templates for the new complementary DNA strands. The first enzyme is primase. Primase comes in and attaches a primer, which are known as RNA nucleotides: G with C, A with U, and C with G. It does this by attaching one RNA nucleotide at a time to the 3' end. This allows for DNA polymerase III to come in and attach DNA nucleotides, A with T, one at a time from the 3' end. This forms phosphodiester bonds between the existing nucleotides. After DNA polymerase III does its job, DNA polymerase I comes in and replaces all of the existing RNA nucleotides with DNA nucleotides one at a time from the 3' end. And that's it for the leading strand. Now we are going to talk about the lagging strand, which synthesizes discontinuously from the 3' to 5' region in fragments known as Okazaki fragments. The first step is primase coming in, binding to the strand, and attaching RNA nucleotides. This allows for, one at a time from the 3' end. This allows for DNA polymerase III to come back in and attach DNA nucleotides, forming phosphodiester bonds This is known as Okazaki fragment 1. Now the DNA polymerase III detaches where the primer is set into place. When the primase sets the primer, which is prime, DNA polymerase III comes back in and sets down DNA nucleotides: G with C, C with G, and A with T. This is known as Okazaki fragment 2. Next, DNA polymerase I comes in and replaces all of the RNA nucleotides, one at a time, attaching from the 3' end. And it does it for the other Okazaki fragment also. G with C and replaces A with T. Last but not least, we have the enzyme ligase. Ligase comes and forms a bond between the newest DNA, which was placed down by DNA polymerase I, and the already existing DNA fragment. And that's it for DNA replication.